Hi everyone, we're the Mikhail group, and today we will be presenting our research which was to study the effect of Eugenol and its analogs on the opportunistic human pathogen Alternaria alternata. This is our outline for today. The overall purpose of our project was to determine which analog of the essential oil, eugenol, is the most effective in inhibiting the growth of Alternaria alternata, which could help in developing a novel and natural antifungal drug in the future. Due to their natural properties as oils, they can serve as better alternatives to current manufactured antifungal drugs, which most commonly lead to drug resistance.
Invasive fungal infections remain a looming problem despite advancing medical practices, and this is due to the emergence of resistant fungal strains, which limit the available treatment that is actually effective and successful. And despite the technological improvements in the detection of these molds, immunosuppressive treatments like chemotherapy cause severe and prolonged neutropenia, which is a major risk factor in developing these fungal infections. On this slide, you can see examples of Alternaria alternata diseases, one of the most common being asthma. The reason why Alternaria is so dangerous is because it can produce over 70 mycotoxins, many of which are harmful to animals and humans. These toxins have been related to hematological disorders and esophageal cancer. Going over eugenol, this is why scientists start to look for natural resources as alternative drugs. Essential oils have antifungal, antitoxic, and antibacterial properties. And when they're compared to synthetic drugs, the fungi will not be able to develop resistance to essential oils over a period of time, and essential oils are safe for humans. However, it's important to note that eugenol in high concentrations could have cytotoxic properties. The FDA and WHO specified uptake limit per day up to 2.5 milligrams per kilogram of body weight for humans. And analogs. Analogs are essentially derivatives synthesized from the original essential oil, which in this case is eugenol. Derivatives like analogs have been found to possess huge biological activities.
For our first method, we did the sensitivity test, also known as the distillation method or the Kirby-Boer test, and these were the analogs that we used for the sensitivity test. The purpose of this was to determine if the analogs used still have the same antifungal effect on Alternaria alternata as eugenol and how effective they were compared to eugenol. This experiment was done twice. For the first one, the final concentration of the analogs was 1,000 parts per milliliter or ppm, and in the second experiment, the final concentration was 250 ppm. A spore suspension was prepared, and a hemocytometer was used to count the number of spores in each plate. The analog disc was placed in the center of CDA Petri plates, which were previously inoculated on the surface agar with 0.5 milliliters of the fungal spore suspension. The plates were then incubated, and the measurements of the inhibition zone were taken on days 3, 5, and 7. The second method we did was the radial growth method, and the selected analogs were taken based on the results from the sensitivity test. The selected analogs were fluorobenzoyl chloride eugenol, bromobenzoyl chloride isoeugenol, and fluorobenzoyl chloride isoeugenol. The used analogs concentrations ranged from 150 to 1,000 parts per milliliter, and in this experiment, we used 1.5 millimeter fungal discs, and we placed them in each plate. The daily measurements of the fungal growth were taken at each concentration for 7 days and compared to the control, which were basically 2 plates with no oil treatment. And the measurements were taken in 3 diagonal ways, and we calculated the average of these measurements to graph our results.
We began our experiment with several eugenol analogs, and the goal of the first diffusion method was to eliminate the analogs that were not as effective. So according to the inhibition zone diameter as shown in this table, the 3 analogs that were the most effective were fluorobenzoyl chloride eugenol, bromobenzoyl chloride isoeugenol, and fluorobenzoyl chloride isoeugenol. These graphs show the effectiveness of our top 3 analogs from disc diffusion experiment number 1. Each day, the fungal growth increased at a smaller rate than control, as was expected. In experiment 2, we used concentrations that ranged from 400 ppm to 1,000 ppm, which were higher than the last experiment. As the concentration increased, the fungal growth continued to decrease, but we still did not manage to reach the minimum inhibitory concentration. In the end, fluorobenzoyl chloride eugenol inhibited the fungal growth by 30.8% at a concentration of 1,000 ppm. This was the greatest inhibition so far, so that analog is the most effective in our experiment. Second most effective was bromobenzoyl chloride isoeugenol with a 27% inhibition, and lastly, fluorobenzoyl chloride isoeugenol with a 25.4 inhibition. Our goal was to know what concentration does the fungus completely stop growing, which needs more experimentation to determine. Fungal inhibition is an important area of study as current fungicides can be harmful to human health. Natural products are receiving more and more attention as potential alternatives to these harmful chemicals, which can often be applied to human skin. Thus, these analogs are safer versions of fungicides specifically able to treat infections. Our results may serve as a guide for future in vivo studies of clinical use of eugenol analogs in treating human invasive fungal diseases. It is also important to consider how analogs can be improved to better target fungal growth in the future. For future work, we need to continue experimentation to determine the minimum inhibitory concentration. We also need to study this inhibition and determine whether it is a fungostatic or fungicidal effect. A fungostatic effect is inhibiting the growth of fungus without killing it, while a fungicidal effect is killing the fungus, resulting in no more growth. After, we would study the analogs, study if the analogs disable the fungal cell wall or the fungal cell membrane using a sorbitol protection assay, which analyzes the effect of the cell wall, and an ergostatal effect assay, looking at the cell membrane. Finally, we would do a cytotoxicity test to determine if those analogs have any lethal effect on human cells. For acknowledgements, we would like to thank Ms. McHale for her time and effort guiding us through our research, as well as the faculty and staff at Fremont STEM for our lab guidance. We also want to thank Edward and his group for making our analogs, and finally, we would like to thank ASDRP as a whole for giving us the opportunity to delve into the captivating world of science.